Researchers at NC State University are developing cutting-edge technology to detect and track plant pathogens. With help from the NC State Plant Sciences Initiative, the Plant Aid project is focused on creating smartphone-based sensors that will detect plant pathogens early and feed the information into a database to monitor diseases across the state. The project is focusing on 4 tomato diseases to develop the sensors: early blight, late blight, bacterial spot, and tomato spotted wilt virus. In the first phase of the project, 2 distributable sensor platforms with a smartphone interface were designed for early diagnosis of pathogens. One sensor measures the emission of volatile organic compounds, or VOCs, from the plant. VOCs are chemical compounds released from a plant that diffuse into the air, typically in response to stress. The sensor will detect these compounds and create a profile that functions like a fingerprint for the plant. Healthy plants will have a distinct profile, as will plants infected by each different pathogen. Comparing these profiles will determine which disease is present on the plant, or if the plant is healthy. The other sensor is a DNA-based sensor that uses a liquid sample obtained through in-field extraction with a microneedle patch to run a test to determine if the DNA of a target pathogen is present on the leaf. The microneedle patch uses tiny needles to pierce the leaf and extract DNA. The needles are then washed and the resulting sample is loaded into the sensor cartridge. The sensor uses a lamp test, or a loop-mediated isothermal amplification. It's a test that can detect specific nucleic acid sequences, including those of plant pathogens. If the pathogen is present, the sample will change color on the display screen of the sensor. The goal of Plant Aid is to first develop and beta test the sensor technology in North Carolina tomato fields at the Mountain Horticulture Crop Research and Extension Center. Eventually, we're going to build a robust database of pathogen presence that can be scaled and used to track spread across the state. These sensors will add valuable tools to the toolbox of researchers and extension agents so they're able to implement more effective management plans for tomato diseases in the future.